Hello everyone. So great to see you today on this beautiful Monday. I hope the weather is fine for you. So today is the second part of our level 2 training. And you're going to always find, like usual, the link for the resource on each of the slides. So today we're going to talk about visualization and figure design, because these are very important when we want to work reproducibly and we want to show other people reproducibly what we are actually doing. So the objectives for today are, I hope I can help you understand why data visualization is very important for reproducibility. And then I hope you are able to learn how to create informative figures that enable the readers to understand your research better. So this is what we're going to talk about today. So let's go into data visualization and analysis. So data presentation is really the foundation of our collective scientific knowledge, because as humans, you probably know that from yourself, we learn so quick and easy from any kind of visualization and figures, right? And this is why figures are especially important, because they can convey a message so easily. I mean, here you can see on the picture of that building. So when you look at that, you instantly will realize, okay, something's wrong with that picture. No one with that building, no one has to tell you that, right? So a figure often shows the data for the key findings. And so what an effective figure should do is it should immediately convey the information about the study design that you used to come to that figure. So if you did, for example, a longitudinal study where you sampled a population, like each week you did a measurement, then the study design would be this longitudinal study, and you would have to use like a line graph. So you have the date or the time on the x-axis, and then the y-axis would be the response of the population. And then people can instantly see, okay, so they measured over consecutive time something. Yeah, so that's a good idea to have a good visualization. Each figure should illustrate an important finding. So it's usually best to have one message and one figure for that. So not to have 3 key messages and try to pack them all in one figure, but have one message for one figure. And if you have done a larger study and maybe you have 5 big outcomes, maybe it's a good idea to decide, okay, so which of these should really be a figure, and how I'm going to present that. I'm going to do the data visualization. Will I work with an image? And in order to not confuse your reader, really try to stick to one message per figure. Ideally, an effective figure, when you visualize your data, is able to allow the reader to critically evaluate the data. So you really should show your data, and we're going to talk about how to do that. So when you collect data, many of us, including me, we use spreadsheet software to type our data in. And that's completely fine. We have to be aware, though, that there are some flaws with this. First of all, it's not very reproducible when we type data into a spreadsheet software, because then when we make any kind of errors, it can be that this error is going unnoticed, and then you're working with the wrong data. Yeah, so if you do that, if you type any kind of raw data from the sheets of papers you have into any kind of software spreadsheet, make sure that the data is correct when you save it, so your raw data is really as it's supposed to be. Sometimes there are something like SOPs, standard operating procedures, or some sort of standardized ways of recording data, or maybe some machinery does that for you. Again, make sure that the data are correct for that. Another thing with spreadsheet software is that if you, for example, use Microsoft Excel, there was a paper in 2016 where it became clear that Excel, for example, is renaming some of the data you have in your spreadsheet. So here it was Excel renaming gene names. So what the authors did is they did something like a meta research study. They scanned the leading genomic journals, so really big names in the world of genomes. And they found that 20% of the papers contained gene lists that had errors in them, where there were some wrong indications. For example, what Axel does is there was a gene name that's called, let's say, OCT18, can be converted from Axel into October 2018, right? Which is not the right name for the gene. And so these kind of things can happen when you use spreadsheet software. So you should be aware that these kind of things can exist. And I'm going to show you from my own research an example. The third issue with spreadsheet software is that the default visualization options are often something like bar charts or line plots, which are not always the best ways of visualizing your data. So here what you see is that's in German because I'm German. You see that's a part of an Excel spreadsheet. So here I was working with a pathogen and I was recording biomasses. So biomasses of plants, like something like 16.77 gram. But as you see, obviously I had something like 8.35 gram, and Excel actually converted this number into a date. Yeah, and so you see that this is only part of the whole thing, but you see this doesn't make sense anymore at all. Yeah. So these kind of things actually really happen. And I was a little bit kind of glad it happened, because then I can screenshot it for you guys and show you that these kind of things happen. But of course I was very bummed out, and I thought, oh my gosh, now I have to kind of write it again. So yeah, of course, I'm also using spreadsheet software. It's just for you to be aware that these kind of things exist. And you can prevent it when you are converting, or when you're saving your spreadsheets into a CSV format, for example, this comma separated value one. And when you don't make mistakes while opening it. So I think I opened this kind of spreadsheet on another computer, and then Excel was like, oh yeah, that looks like a date spreadsheet, and it just did that, right? So just be aware. So the interpretation that we have on any kind of visualized data depends, of course, on what we see, right? So here we have two different ways of presenting data, which actually it's the same data, it's just two different ways. And you can see here, oops, on the left, you see these are the bar graphs I was talking about earlier. And people here also use the wrong statistics. So that's the average, the mean. And then this little error bar here obviously it says SE, standard error of the mean. It should be the standard deviation. But yeah, this is what we see here. So we could think, okay, so in the second group, maybe that was like the maximum response. Here on that hand, we as readers are the active participants. So you can see these are the raw data, yeah? So we have these four groups, and if we want to make a bar graph out of it, this is what we come up with. But you can see here, there's some issues with this kind of data set. First of all, it's only a few observations. So here it's like 5, here it's only 3. Here we have 6, and then here we have 6 again. Here we have an outlier. And of course, if you present the mean or the average, an outlier will greatly disturb that average. Yeah. So you can see here the average is up here because of that outlier. Here what we see, this is a dot plot, and this dot plot here has as a summary statistic the median. The median is cutting all of the data points in half. 50% is below the median and 50% is above it. So you see that this way, the right one would be a much more transparent, reproducible way for the reader, because here as a reader, you're an active participant in the data analysis, so to say. So what we want to do is we want to avoid bar charts for continuous data. Continuous data are data that can have any value, basically. They can be below 0, they can be above 0, they can have a comma or not. So any kind of data, like these biomass measurements that I've shown you. And here you see we have, of course, different data distributions. Yeah, so here we would have a symmetric kind of population response. So maybe it's a similar amount of data that we have recorded. And if we do the statistics here, for example, we do the t-tests or the Wilcoxon test, you see these are the calculated p-values. Here we have an outlier, and again, when I display this one, number C here, as a bar chart, I get exactly the same bar chart, yeah? So all of these different data distributions here lead to the same bar graph or bar chart, yeah? And that's, as you can see, a problem. Because here we have an outlier, which is pushing the median, the average a little further up here. So that would not be a very transparent way of presenting it. And you can see here the p-values, of course, change. So when I compare these two populations, I have a different outcome in the calculated p-values. Here we would have a B-model distribution. So maybe we have observed female and male mice, for example, and maybe the male show a different response than the females, or maybe there's some other kind of stuff that you are seeing. And again, here, it's not a good representation in a bar graph, right? And then here we have an unequal amount of sample sizes. So again, not a transparent way. So we don't want to use bar charts, although Excel or other software is suggesting that. We don't want to use that for our data, specifically with a small data set. So really, there's no way around. We should really show our data as good as we can, because we don't want to hide behind the summary statistics. Yeah, so this slide, I use it for teaching as well. And you can see the data points above here, they would be invisible, right? Here, they would just hide behind the bar. So I hope that I convinced you already that it's not a good idea to trust the summary statistics alone. If I haven't convinced you yet, I think I'm going to with this scatter plot here. So we have a scatter plot, and then we have a predefined x and y, and the standard deviation for that and a correlation coefficient. Now, if I run this kind of simulation, it's called the Datasaurus, you see that all of these different data points in space, although they have the same x and y coordinates and the same standard deviation, all of them look very different, right? So if you would just present the summary statistics, we could have anything in here in this kind of scatter plot, right? So really do not rely only on the summary statistics, but really try to show your actual data. So what do we do? How do we choose the right plot? So if we have continuous data you have a small data set, and the majority of researchers are using small data sets and they're very valuable, it's a good idea to use a dot plot like this one to really show each of the different points here. And then you could have, as a summary statistic, you can have the median, for example, here to show that. You can also, of course, show the interquartile range or well, yeah, whatever kind of thing makes sense for the context here. And this goes for any data distribution. If your data set is a little larger, I would typically say something like above 15, a sample size of 15 or more. A dot plot can get a little bit tricky depending on how close together your data points are. You could use a box plot and display the points. The box plot gives you the summary statistics alone already. You would have these quartiles. You would have the whiskers here that show basically the min-max in a way. And then everything outside of the whiskers would be an outlier, very likely. Of course, you would have to do outlier analysis to determine that. But they give you a good kind of overview of the data. If you have a large data set where it doesn't make sense to show the individual data points, you could just use the box plot as it is on its own, or you could use a violin plot. Here we have the option to show the points or not. And here you can see the data distribution also. And so a bar graph really should be reserved for only the cases of counts. So when you have really counted something, so it has to be something above 0, and like a whole number, not any kind of fraction, or a proportion. So I'm working in a molecular biology, so I often have something like PCR data. And so the relative expression of a gene, for example, is kind of relative proportion that's often shown as a bar graph. So these kind of cases, they are fine to use a bar graph, but if you can, try to use another option. So how do you make an effective dot plot? Here are a few tips. If you really want to put the emphasis on the points, you want to make all of the points visible, it's a good idea to not put them all above each other, but actually to play around a little bit with the dot size. Sometimes these kind of semi-transparent points make a lot of sense, or you can use the jitter option. And I'm showing you a tool where you can do these kind of dot plots on a slide coming up. So random jitter or symmetric jitter, whatever you prefer, would be a good option. So here I think we can conclude that this is kind of a nice way of showing all the data. And I think these are these are definitely more than 10 data points, and they are still visible. So you can still see, okay, the majority of the data are here, and then we have a few of these responses that are a little bit outside of the norm, it seems. If you want to emphasize, or after you have concluded how to show your data points, then you also want to add some summary statistics. Maybe you want to emphasize the summary statistics alone because you want to show, okay, here, for example, I have like these 5 different or 6 different groups, and I want to show the differences, and people should focus on the summary statistics. I'm showing the data points, but the summary statistic is one I'm going to talk about. Then it's a good idea here, instead of doing this kind of cramped figure, to play around a little bit with the axis here to increase the width a little. And then you would emphasize summary statistics here by making the plots again, giving them like a little bit of a color, but getting rid of this kind of outline of the dots. And so there is an interactive free tool you can use, interactive dot plot. It's hidden under this link here, and this is what you can use for making dot plots. I use it in my teaching. My students are using that. It's relatively easy to use. What you do is you just you upload your data, you define your data set. So you have to read a little bit through the instructions, but it's pretty easy. You have to just format your data in a certain way, upload them as a CSV file, and then you can really play around here. So you have all kind of options. What do you want to show? Do you want to have a dot plot, a box plot, violin plot, all these kind of things? Yeah, and then you could have like also subgroups if you want to. You can color code them, and then you can export these graphs. And then of course you could also like use another kind of software to make them a little more pretty. But I would really recommend that you play around with this. If you have a lot of different groups that you compare, I think that tool is restricted to a certain amount of groups. I'm not sure, maybe 6 comparisons or something. I can also write in the Etherpad another tool, another 2 websites I'm using also to do these kind of dot plots. So doing them with Excel is really a pain, or any kind of spreadsheet software. If you know how to use R, that's a very good idea, because most of these tools are actually depending on R. They are basically running on R, and then people made like a graphical user interface. Yeah. So using R, of course, is also a great option. The ggplot package, for example, you can make really nice visualizations. I'm a dinosaur. I'm not really doing that. I'm using these kind of websites to do that because I don't really need it so often in my work. Here's an interactive line graph tool. That's also very useful. Again, that's something I do with my students. And they use that when we have like this kind of longitudinal study. We have a nice way of presenting these kind of observations, also with some summary statistics, which are, again, really hard to do in Excel or something. Yeah, and then there are some other options. I think that slide is a little bit outdated because I think it's not Prism 8 anymore. It's already a little bit further. So these are also options. And I have to say, I didn't check if they are still free. They were supposed to be free of use. So these are a bit more fancy options also, if you want to have something, yeah, that's a spreadsheet software, but a little bit advanced probably. All right, so let me check the time. We would have, I think we have time for Prism is not free anymore. Yeah, we did this slide in 2018. We're currently revising our slides. I'm usually having the updated slide in my own talks. I just didn't have the time to do that for today. But yeah, Prism is not free anymore. It used to be. But yeah, as I said, I'm going to post some links, two more links of stuff that I'm using also, which are all browser-based free tools. Okay, do we have any kind of question already? So if yes, please put that in the Etherpad. Or you could maybe for one question unmute yourself before I continue. So this is the link to the Etherpad again.
Susann, I have a question.
Yeah.
I think one of the things that make me sort of lazy and use Excel, for example, is that you could sort of ask it for suggestion on which graph type that better describes your your data. So are there any like sort of software or tools that like would, I don't know, give you some clue about which type of plot would like be best for your data, or you have to study that and do it on your own?
So I think Lamas, as often there's no definite answer to that. I personally like the decision matrix that I've shown you, where you, sorry for the run, where you really, I would always suggest to really try to make it as easy as possible, even if it's boring. There are always new ways of displaying stuff, and people always like to jump on a new trend, volcano plot, and this plot and that plot. But for many of the options nowadays, you need more kind of, the reader needs to kind of learn a little bit first how to read and interpret the plot before they're able to see your data, which I think can be a disadvantage. So if you are working with continuous data, I think these options are, even if they are boring to some people, people are easy, like they are accustomed to read a box plot, for example, oftentimes, hopefully. And if not, you can quickly look it up on Wikipedia. So I would personally try to stick to these if you're working with continuous data. If you work with data, let's say any kind of like nominal data or like scale stuff, like I'm a plant pathologist, so I'm rating plant diseases, and then I have a scale of something like 0 to 5. Here, what I'm doing is actually when I show my disease ratings, I'm actually working with bar graphs, with stacked bar graphs to show the proportion of how many of my plants are in which category. So I would have like a bar graph, but at 100%, but each bar is divided into sections, color-coded, usually with grayscale, to show how much of my plants was in each category. And then because my disease ratings, the outcome of my disease ratings is a number, like a count, this is the way that in my field people display that kind of disease rating outcome. So that would be a bar graph, because it's like one number, right? I could use a dot here as well, but that usually doesn't really make a lot of sense. So there's no definite, I don't know about a software that does that. I think depending on your field, I mean, in your field, you're reading papers and you see what other people are using. And if you like a paper for its easy display of how they present the data, then I would like be inspired by that and maybe try to use that as well. And if I cannot do that, again, I would refer back to the very simple options. I think the key on what I would try to really come across here is, do not make it overcomplicated. Really try to go with the easy way of doing something, and then not like make it too complicated. So really, if you show your original data here as a dot plot, then that's it. That's how you show your data, and everyone will be happy. Yeah. So, but yeah, we can, if I have a bit more time, we can also explore the other options, what I can show you then as something like a, yeah, what I'm doing. I just have to dig up the link for you guys. Okay, I hope that was a little bit helpful. Let's go into designing figures with images. So we talked about data visualization. So you have counts, proportions, whatever. Now you maybe have some pictures, some images you took of machinery or of your pets, animals, whatever, plants, whatever kind of specimen stuff you work with, or yeah, whatever you can think of, and you have to display that. So let's just first try to understand why it matters so much to make a good figure. So just like with the data visualization, where we like to look at figures first, you really have to be aware that often scientists, I mean, all of us, we don't have a lot of time. So when we come across a new paper, the first thing we often do, and also reviewers do it, editors do it, they examine the figures first. So nowadays, when I go and I search for papers I use for my teaching, I usually, the first thing I do is I read, like, I skim the abstract if the topic is what I want. And then I look if there are any kind of figures, because for teaching, figures is like usually what's most important and what's useful. So figures should really be accessible to a broad audience, because you never know who's probably using your outcome, the figure you created at some point. Also, you talked about open access. Even if you have a closed access paper, search engines like Google, if you do a Google photo search or picture search, image search, and also journal websites usually allow people to examine figures of papers, even if the rest of the text is behind a paywall. So the figure will be very accessible in a way if you go to the journal website, right? And then many of us are now, if we are proud of our accomplishments, we share our image-based figures on posters, of course, on conferences, and maybe in social media. So a figure is really important, and it should be a good one. So try to keep that in mind when you design the next figure, that it's maybe not only for your personal use, but if you do a good job, you can share it really widely with others as well. And that brings us to the audience. So things that are clear to you may be confusing for readers from other fields, and maybe even also for people that are working together with you, sitting just across from you on the other side of the desk. So think about designing your figures for a broader audience so they are more easy to understand, because your readers could not only include scientists in your field when you do a poster presentation at a conference, but maybe scientists from other fields, reviewers, educators, grant officers, people like me who do a lot of teaching. We skim the papers for good figures to use in our teaching, because we don't want to have like a lot of text usually. So it's a good idea to ensure that your figures are self-explanatory. And there's a good tip from Ahmed, if you code in Python, to try matplotlib and Seaborn. Ahmed, can you put these links, or someone else, please put them in the Etherpad? That would be very useful. So that was for the previous part. Thanks a lot. So the stuff I'm presenting now in the next couple of minutes actually comes from a work I did together with colleagues. Most of us were eLife ambassadors as well. So we were in a previous cohort at some point. And we did a meta research project where we looked at, like, we downloaded thousands of images from our own fields. So for me, it was plant biology, others used cell biology, and we used physiology. And what we did is we did like a rating on the pictures, on these image-based figures, on how accessible they were, if they were colorblind, were they easy to understand, was there enough context provided, and we basically rated them through a certain scheme, so you can read that in the paper itself. And then we came up with some suggestions to create clear and informative image-based figures for any kind of scientific publication. And so we were in the meta research project, you always have these kind of, let's call them reviewers. You basically control the work of the others also. So you always are making sure that the quality, the ratings you do are the sufficient quality. And the outcome for my field, plant biology, was really kind of drastic, because we found that only 2% of all the figures that were in plant biology, and in plant biology, you have like microscopy images, you have plant images. So anything you can document on a plant, any kind of tissue from a plant, only 2% of the people used a scale. Yeah, so we had all of these pictures with plants, but there was a scale on only something like 2% of them. And so, I mean, you know how plants around you grow, right? I mean, a tree, for example, of course, it's a large plant, but maybe a tree can be small, it can be bigger. So it's really important to have that scale information to understand what you have actually researched here. And so what I did when the paper came out is I posted this on Twitter back in the day, a couple of years ago, just to show how our perception can really be distorted. So I'm working with Petri dishes. Petri dishes are usually 9 centimeters, like the standard sizes, but there are Petri dishes that are bigger or smaller. And what you see here is a part of a Petri dish where I was growing my plants on. So these are plants, and they, for someone who's working with the same plants, that looks like these plants are very small if they think it's a normal sized Petri dish. But here, as you can see, I took a picture of my plants and also of the scale. So what we see here, we have this black and white kind of thing in the background. And this is a scale of a centimeter of each. So it's basically looks like a ruler, and like the white part here is 1 centimeter in length, and then the black is a centimeter, and so on and so forth. And so you can see that this is not a normal sized small Petri dish, but it's a larger one. And that's why it's so important to have a scale on that image. And that's like one of the things that I'm teaching and preaching to all of my students right now, because it's so important to have that. So here are it's a list of tips basically when you prepare image-based figures, and then we're going to go through some of them a bit more in detail. So first of all, you should choose a magnification and a scale that fits your research question. So if you want to present teeny tiny details, of course it should be, you should have magnified images of what you want to present. You should, of course, always add a clearly labeled scale bar, something most people don't use. Use color wisely, choose a colorblind, accessible color palette if you can, and we talk about why that is. When you make your figure, before you actually do anything in any kind of software, it's a good idea just with pen and paper to design the figure layout as a sketch or so, or maybe make like a table. And in that paper, there are some tips on how to do that. You should annotate the figure. So the figure should have labels and annotations to make it really accessible to a broad audience. So someone can just look at your figure and all the labels are there, and they don't need to read any kind of other stuff to understand your figure. And then, of course, when you prepare legends, you need to have clear explanations. So here's a step-by-step guide that you can also find as a teaching resource on that paper. So the link to the teaching resource is here. It's on an OSF repository where you can find these slides together with the paper versions. So choose a magnification and a zoom that emphasizes the key message of your figure. Like with data visualization, you should limit yourself to one key message per figure. So here we are in a microscopy context. Here you want to show, okay, so we have a nucleus of, so the core of a cell. Here it's a muscle cell, and we want to show that the envelope, so something that's around here, the nucleus, in the mutant is different from the wild type. So that the scale, of course, would be like we would zoom in into the nucleus and provide that kind of information. Here's a scale bar. It's not labeled because in this case, we want to show the difference between both of these, but then ideally that scale bar needs to be labeled as well, of course. And the key message would be here, okay, only the envelope of that nuclei is different. So this is what we show next to each other. Maybe if you want to zoom into the image and provide more detail of the image, you could use an inset. And an inset is basically just a magnified version of a part of the image. So here you would have a microscope image again, and maybe you want to enlarge this part here. So you do that by showing it next to that microscopy image. And then it's pretty good to see, okay, so you magnify this part, and then you kind of have like this zoom out kind of lens thing. And then this is the magnified part of that image. Yeah, and this kind of thing is called inset. Here you have from archaeology, you have, as you can see, some bone remains. And then again, we have an inset here because this part of the image is enlarged again here. And you can also see the scale bar here. So that's 1 centimeter then here. So when you use these kind of insets, which can be really helpful. Make sure that you accurately mark them inside of the original image, and that you clearly explain in the label attached in the legend, in the figure legend, what the inset actually shows. Step 2, decide whether to use color. If we are back in, so if we're in the microscopy world, or maybe also in other worlds, but let's go with that for an example. Some of the images are already black and white because an electron micrograph is black and white. So of course you use black and white because that's the natural color. If color conveys some important information about the natural appearance of some subject that you investigate, then of course, maybe it's a good idea to use color. Like here we have this kind of Drosophila or some other fly picture, and maybe here we want to show, okay, they have like red eyes. So of course, then you would use a color image, right? If in microscopy you work with any kind of staining or fluorescence and you only have this one color, so you have a sample that is fluorescent and the other one is not, you can use color, but again, you should consider using black and white because the contrast in these images is actually clearer and grayscale. So here we have our wild type mutant picture again. This is how it looks like under the microscope. So because our filter and maybe our staining makes this part of the tissue red. You see here red and black, they are really hard to distinguish. So here it would be a better option to convert that image into black and white because this is easier for our eyes to see. Our eyes are able to pick up a lot of different grayscale variations. I think something like 20,000 different shades of gray. So it's really a good option to actually consider that as a color, so to say. If you have any kind of staining or fluorescence and you have more than one color, then using color, of course, is usually a good option to make it clear. But white is also still a color you could use. So here are some examples. Here we have a number of microscope images. You can like convert them back and forth as you see fit usually. Here we have an example of good visibility or really best visibility. So we have a black background, and then we have either this kind of yellow cyan, which is this kind of bluish color, or green on black. And they are all pretty good to see, but we can also convert them into grayscale and it would still be fine to see, right? What is not giving a good visibility is magenta, red, and blue. So anything with a bit of a orange or like red kind of undertone, and black is usually giving a really bad contrast, specifically black and blue. These are just really hard to see, as you can see here. So using yellow, cyan, and green is a good idea if you want to use color. Now, step 3, test if your image is accessible for colorblind readers. If you use a software for your image preparation like Fiji has, I mean, this is like a previous version of Fiji, but it will have at some point, will have this kind of option color and simulate color blindness. And then it gives you kind of an image of how it would look like for a colorblind person. I mean, we know, probably all of us know that green and red are bad combinations because that excludes colorblind people from being able to distinguish. But you could use magenta, green, and blue, or magenta, yellow, and cyan, or red, cyan, and yellow as safe options. And of course, again, white is a good option. So the most common form of color blindness is deuteranopia, where people who have this kind of condition, and that's in Europe, that's something like 8% of the population. It's mostly males, so mostly men are affected. I'm not sure how it's on the African continent. In the US, it's pretty similar. So something like 8% of the population has it. And a person with deuteranopia would not be able to see that this is red here. I'm not sure if you would see my red dot here. Very good. So we have red and green here. We have a little gecko sitting here. And that gecko is green, and people would not be able to see that plant here probably with the gecko together. And then tritanopia is another condition. It's more rare. where people then have issues in general of seeing green, for example. So it's actually likely because of how often this occurs that some of your co-authors, reviewers, editors will be colorblind. And many of the readers who are reading your paper will be colorblind as well. So it's just a good idea to make your pictures colorblind safe, because then it's like it benefits everyone, also normal sighted people. And if you want to test for colorblind accessibility, you can download a free small tool, Color Oracle. It's a very small tool that you download. And then when you're working on your computer and you have on your screen the picture you want to test, you click on this little Color Oracle icon, and then it transforms your picture or what you have on your desktop into the Deuteranopia and Tritanopia mode, basically. So you can see how a colorblind person would see that image, and this is how you can test for colorblind accessibility. There's more information also in the paper. All right, choose a labeling style. So I said that you should annotate and label your pictures. So again, if we're here in the microscopy world, there are different ways you can label. I'm not going to go in detail here, but of course you can use lines to label something to make it clear what is what. There are no limitations usually. You just need some space below the image or next to the image that allow for you to do this kind of labeling. And of course, the lines should not cross over. You could use arrows. Arrows are actually good and helpful if you want to show direction. So you could show direction. For example, here we have a nerve cell, and then these kind of nerve cells are migrating at some point in this direction. And I think, if I'm not mistaken, they go in this direction probably. So you could like have like a dotted arrow or something to show movement. You just have to really make sure in the legend that you also describe what's going on here. And then you can also use symbols or letters or something if you have a lot of stuff to label in your image. The advantage here is it's good for having many structures labeled, but then the reader always has to go back and forth. So that's a bit of a disadvantage here. However, you should make sure to not conceal the original image here. And if needed, that's a bit more fancy. You could put color on top of your black and white image. So superimpose that to show specific structures here. That's another option. That requires some detailed work and quite some time to do it correctly. All right, step 5, every image needs a clearly visible and labeled scale bar. When you have microscopy software, the software often comes with an automatically labeled scale bar. But if you cram 10 pictures of your microscopy into a small figure, that scale bar will be so small that no one is able at 100% to read that scale bar or even identify it. Yeah, so make sure that even if your software is already giving you a scale bar, you actually either you adjust it a little or you just draw a new one on top of that. And it should always be labeled, because then you cannot forget. That's something people do in the legend. They forget to write what is the size of the scale. Yeah, they just forget it. They have all that fancy picture and then they forget to label the scale bar. So just do it in the image. It's much easier to do it this way. Yeah, to drive the point home, every image needs a scale bar. Even if you take a picture of machinery, like you take a picture of your computer, it still needs some sort of scale information next to that, right? I've seen pictures of people working with rats or mice, and they have different sizes. It's not like each rat is the same size. Again, you need always a scale. And I mean, you could put like a ruler next to that. Yeah, so something like this here. This ruler here has this black and white kind of thing. So each centimeter here is like black and white. It's like an alternating pattern. So even that would be helpful, right? And maybe don't use the banana for scale. I know that's like a meme, but we're on science here, so an actual ruler would be probably a little better. All right, Step number 6, write a figure legend that can be understood regardless of having to read your paper. So the figure legend and the figure itself, they should be standalone. Yeah. So maybe you have like a little bit of methodology there to put it in context. So all these kind of things give you readers that are very happy with your paper, and your paper is easier to understand. And then the last step, and that would be really cool if you could do that, is to test your figures and legends out on other people. So can a friend in the neighboring lab who know kind of what you do easily understand the key message? Get feedback from your peers, yeah? So that's always very valuable. And try to get that feedback before you send your paper to submission to review. All right, so much of that work was by my fabulous colleague, Helena. She has written a lot of papers. She's a data visualization wizard, so she has great ideas how to do these kind of things. I just linked 2 recent papers that she has published, like here 10 simple rules for designing graphical abstracts, for example, or insights into poster preparations. And I hope that these are helpful for you. So please check out more of her work. Yeah, and then we summarize by how we always do it. Reproducibility is, you can see, it's a wide scope. We covered a lot of different things already in these trainings. I hope you are able to take some of the things in your own research. Here, the part that we talked about today is hopefully helping you to, well, accurately analyze results, as in like accurately display them to make good sharing possible, so other people are able to see how was your experimental design for that figure, or when you have images in your figures, what was the scale of that? Is this like a normal size, or is this like one of your key messages, like, oh my God, I had these monster plans. Look at the scale here, right? How big these were with these treatments. So all of that plays into making stuff reproducible, making research reproducible. So I'm interested in you taking the quiz, where I'm going to ask you also what you're going to do and probably change when you create your next figures. And the quiz, I'm going to share that also. So this is like the quiz for the level 2 training. I'm going to share that link in the chat also. This is the quiz that you please take to complete your level 2 training. And I think I'm going to stop sharing now. And then we can talk a little bit because we have some time left. So thanks a lot for being here. And I'm just sharing that link with you for the quiz. So if you have the time right now, you can do that. And if you have questions, of course, you can also just unmute yourself and put your question in the Zoom chat or in the Etherpad.
Yeah, I think this is the best data visualization session that I've ever had in my life, to be honest. And I wish if like in undergrad or postgrad, we were taught about data visualization like this. I think this is like this should really be incorporated when we teach students about data visualization. Yeah, so thank you very much. And I just wanted to add one thing that I usually do, and I find it helpful, is sort of taking the step of asking a neighboring lab mate about like how effective is the visualization you have. Is I usually go to my husband, you know, he have absolutely no clue about what I'm doing. So I sort of ask him, and when like if he gets the idea, then I'm comfortable, you know, that if this was seen by someone who at least have some clue about what I'm doing, then this would be easy to understand. So this is something that I also recommend doing.
100%. So when I was doing my PhD, I tested a lot of my presentations on my husband. So he also knew what I was doing in too much detail for his liking, I think. But yeah, I mean, I mean, we don't all have to be good science communicators. I personally think it's a good idea to be able to do science communication. And part of what I've shown today is also this kind of science communication aspect, right? To always not design the figure only for yourself, but really think about, okay, so I have these colleagues that have no clue what I'm doing. How can I kind of take them where they are right now and still show them and make my kind of work useful for them? So they provide some context, of course, right? Yeah, and I mean, we shouldn't get distracted by all the fancy kind of ways you can present your data. That's for people who are experienced, I think, but maybe if we just start with the kind of very basic stuff and just make it very visible and transparent, I think that's often really enough, probably.
Yeah, I couldn't agree more, because now I see, like, data visualization is supposed to make the information easier to understand, but now we see all sorts of 3D graphs, and they look really cool, but as you said, like, you need to study how to read the graph before you're able to do it. So yeah, I couldn't agree more. And I think everyone in the chat is also agreeing.
Yeah, I think you have to have a degree in data visualization to understand the plot.
Exactly.
No, no,
Exactly.
sorry, but no. Okay, let's
S-
search for this other tool, but I'm listening to you.
I think Naira, you're unmuted, so you would like to ask a question? Maybe.
No, 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 no. I'm
No
okay.
worries. No worries. So does anyone else have a question to ask for Susan? You can unmute, or you could write in the chat.
So I'm sharing that that other resource. I promise I put it here in the chat first. And then I also put it, I'm just showing you how I use it. Plot options, yeah. Okay, let me dig out this other presentation here. So I'm just going to share my screen again. As I said, I didn't have the time to include it this time, but what I love to do is using rain cloud plots. So maybe you have seen there's a film, a movie, it's called Trolls, as in, I don't know, trolls. And there's the rain cloud guy, the very cool guy. And yeah, so the rain cloud plots, what they are is, this is, it's a Shiny app. So someone used R, it's actually Cedric Scherer, who's a guy who's very talented in making these R options available to dummies like me, who don't like to do a lot of things with R by themselves. So it's a Shiny app. It's a website. I just shared the link to that. And here what you can have is you could have a bigger data set, and then the rain cloud would be, you have all of your data points displayed. And above that, you have half of a violin plot. So the violin plot, if you remember, was this kind of vase-like form, like this, but on the other side as well. And here people have just cut it in half, put a box plot here, and then the data, the original data are next to this. So it's all the same data. You have the dot plot, you have the box plot for the summary statistics, and then you also have the violin plot. For people who don't know what a box plot or violin plot is, that might be a little bit tricky, but you can also adapt the way that this is displayed. And I do this, again, I do this with my students, I do it with my master's students, that this is the way that they can display their data. So it looks like a rain cloud, and then you have like the rain is all your data points. And here you can also see the outliers, the potential outliers. And you have the option to do some statistics with this one. So I like this one a lot. It's like, it has more functionality than in a very simple dot plot. And here you have all kind of options for the colors and everything. So again, it's on R. The R is the basics and ggplot. And then Cedric Scherer took that and basically made it into a really nice, fancy visualization of the data. And it's free as well. There's a paper, I think, to that. So if you are working with R and you want to do it yourself, you can also, there's like a step-by-step manual how to do it yourself. But I prefer this kind of option. So I like this a lot.
Yeah, I like it also. It looks really nice. And as you said, not necessarily all of us would be able to use R to the extent that professionals could. So it's nice to have these tools for dummies, as you said. So it's easy for us to use. Yeah.
Yeah, I mean, you know, I'm always saying that. I say that to other people that I'm a dinosaur. I mean, I had R when I was a student, but I don't use it so often. I don't do an extensive amount of statistics because, as I said before, I'm working in plant pathology. I do like disease ratings, and I don't need all of this. So here's another tool, just before I come to the question, I'm putting that. Estimation statistics, you can click on that link as well. Here what you have, it's another way of displaying the data. And here you can calculate effect sizes. That's what I usually do. I don't do, well, I have a good statistic foundation. I'm a trained ecologist. And many of the stuff I'm doing is not fit for using a t-test or anything, but I'm calculating effect sizes, and that's something you can do with estimation stats. Again, I think it's on R. There's a Nature paper to that. That's also as a preprint available for free to see. So these are tools, you can just use them, and then you cite the people who made the tools, and you know, just get on with life. All right, Thomas, you have a question. Do you want to unmute yourself? Thomas, you have a question? Do you want to unmute yourself?
Oh, no, thank you. It is okay. It was an error.
Okay.
No worries at all. So I think we ran out of time, but just to a final check, does anyone else have a question before we close this session? And I guess if you like watch this session again and you come up with questions, you could always use the Etherpad to post the questions, and hopefully, Susan, if you could check on it maybe a week from now, then you'll definitely have all the questions in there.
100%. And you can, of course, also put stuff in Slack, right? If you have a question to them. Yeah.
Yes. And Susan is with us at the trainees Slack. So I think you could easily reach out to her. Yeah, and as Samira said, this was really the cherry on top of all the reproducibility sessions that we had, because I think each and every one of us could directly apply that to their work or their teaching. So Susan, we cannot thank you enough, to be honest. Having you in this training, level 1 and level 2 have been amazing. And we hope that this is not the last time that we see you. We'll definitely have more things to do with our LNLs in the coming future, and it will be an honor for us to have you there. So thank you very much.
So glad to be here. Yeah, so for the quiz, I'm not sure actually the deadline for the quiz is when you guys are finished with the whole thing. So maybe in 2 weeks. We didn't put a deadline. I'm not sure how many more trainings you have.
Yeah, we have one more training. So I think the deadline will be in 1 week, more or less. So yeah, I hope that all of you could finish up the survey in 1 week. Yeah.
Okay, so I'm posting the link also in the trainees part of Slack, and then I write the deadline for that. Yeah.
Perfect. Thank you very much, Susan, and thank you, everyone. We had a really good crowd today. It's really nice to see all of you here, and yeah, we'll see you again for our trainees in the coming sessions. And thank you, Susan. Thank you very much.
Thank you so much. I hope you have a great week, everyone. Bye.
You too. Bye-bye. Bye.
Bye-bye.
Bye bye.
Thank you very much. Bye.